The overall goal of this approach is the fast and reliable screening for microbial exopolysaccharide producers and the identification of their carbohydrate fingerprint. This is an essential step in order to utilize the unexplored diversity of exopolysaccharides.
Our method can help to speed up the polymer research in the field of microbial exopolysaccharides. It enables the fast identification of novel polysaccharide variants with different properties for specific applications.
The main advantage of this technique is the combination of different exopolysaccharide detection systems in a modular and completely automated screening concept. This makes it extremely fast and reliable.
This technique can also be performed manually in laboratories where there is no automatization platform available. Thereby, its use is very flexible and can be adjusted to different screening needs.
This exopolysaccharide or EPS screening platform has a modular setup, which allows a separation of the protocol into two major parts: automated screening and carbohydrate fingerprinting. The first task is to cultivate the strains for screening and remove the cells by centrifugation. The strains are cultured on glucose as carbon source in 96 well plates covered with breathable sealing film on a microtiter plate shaker at 30 degrees Celsius and 1000 RPM. On the day of the screen, prepare the robot work table and storage carousel as described in the protocol text. Remove the breathable sealing film from the plates and allocate the main cultures in carousel positions 1-1 to 1-4. Start the automated robotic program.